So, good afternoon, everyone. Today, I am going to discuss about the differential susceptibility and role of senescence in CAR-T cells based on their co-stimulatory domains. So, as we all know that despite of the success of CAR-T cells in hematological cancers, the success in solid tumor is still very low. So the key hurdles in the CAR-T therapy is due to the immunosuppressive tumor microenvironment, escapes, and then the off-tumor effect, and then limited CAR-T cell persistence in vivo, and the functional exhaustions. So these factors limit the ability of CAR-T cells to induce long-lasting effect and efficacy. So, in this article, they've reported that CAR-T senescence is a major role in the CAR-T senescence has a potential resistance mechanism, especially in CAR-T cells which have 4-1BB co-stimulated domain. So, to show their results, they mimic the cancer relapse, where they've utilized in vitro model with repeated CAR-T cell activation, which is followed by the rest period. In their study, they have used the FDA-approved CD19 targeted CAR-T cells, where either they have used 4-1BB co-stimulated domain or they've used CD28 domain, where they have shown that CAR-T cells undergo functional, phenotypical, as well as transcriptomic changes of senescence, which they have found out that it was more prominent in the cells which is 4-1BB co-stimulated domains. So, to analyze their studies, they have utilized additional independent studies also to induce senescence. One was through induction of MYC expression, and other was the irradiation. So to go further, let's one see the different generations of CAR-T and how they are different. So as we all know, in first generation CAR-T, it has only that these are antigen binding domain, this is hinge region, this is transmembrane region, and this is transmembrane region without any co-stimulatory domain. Then in second generation CAR-Ts, they have induced the co-stimulatory domain, either 4-1BB or CD28. Then further, next generation CAR-T came, where they have found out, because these co-stimulatory domain, either 4-1BB or CD28, they both have different functionality. So then together they bring the positive of these two different domains into the third generation CAR-T, where they have added both the co-stimulatory domains. Then after, in fourth generation, they have added another protein which is called cytokine inducer fragment, which is only after this is kind of inducible kind of thing. So whenever there is any antigen, only at that time they induce the cytokine released. Then in the fifth generation CAR-T, they have added another, this is called gated CAR-T also, or truck CAR-T also. So in the main role of co-stimulatory domain, as we all know that this is a co-stimulatory domain is an intracellular signaling component, which increase the activation, proliferation, as well as the survival of the CAR-T cells. So now we will understand that what is senescence in CAR-T cells and how it is different from the exhaustion. So senescence is a stage of permanent state of cell cycle arrest, where the T cells loses their ability to proliferate their functionality, even though they are metabolically active. So this phenomenon limits the expansion, persistence, and the anti-tumor efficacy of the CAR-T cells, specifically when there are adverse tumor microenvironments. So the key features of senescent CAR-T cells are loss of proliferation, functional decline, where they've reduced production of cytokines. Then there is upregulations of senescence markers like P16, P21, P53, and beta galactosidase activities. There is altered metabolism also has been seen in these CAR-T cells, where there is higher production of ROS, and mitochondrial dysfunction also has been observed. Then further there is epigenetic reprogramming. With this, there is a T cell exhaustion marker is also has been seen in the senescence cells. So they can overlap together. There can be senescence markers also, and then exhaustion markers also together can be found. So in this study, they perform these experiments. First, they generate the CAR-T cells. After that, they generate the in vitro model for CAR-T cell failure by repeated activation and rest cycles. Then they've done T cell functional assays. Further, they've done experiment in vivo also, where they transplant the cells in NOD-SCID mice. These are immunodeficient mice. And further, they check the efficacy of CAR-T cells in vivo by checking the expansion of the CAR-T cells and cytokine release profile. And the proliferation was done. Proliferation was checked by EDU assays. Then they've isolated RNA sequencing. They've done qPCR, Western blot, as well as analysis of telomerase length. So for to make the this CAR-T cell model, they have taken mantle cell lymphoma cell line JECO1, which was lentivirally transduced with the cassette having luciferase gene. After that, they generate the CAR-T cells. They've generated the second generation CAR-T cells. Either they have used CD28 domain or 41BB co-stimulatory domain. Then to prepare the CAR-T cells, they have isolated the T cell from healthy donors. After that, they added dynabeads in 3:1 ratio. They grew the cells. And then after that, after 1 day, they were transduced with the construct with the multiplicity of infection of 3. That means if they have used 1 million of the cells, in that they have added 3 million of viral vector. After that, the cells were grow in the T cells growth media, and then on the 6th day, the beads were removed and the cells were further rested, and they were called the baseline cells. This is the complete flow chart of the how they prepare this model. So first here we can see they've isolated the T cells. After that, they've added dynabeads, CD3 CD28 dynabeads to it, and then the cells were transduced. After that, the beads were removed. The cells were either freeze down or they were used for further activation. So these are the day 8 cells they have used as a baseline cells. These are the cells which are like regular CAR-T cells. So to make their model, they have activated these CAR-T cells with JECO-R1 cells which were irradiated. So for 3 days, the cells were activated in the presence of JECO1 cells, and after that they were in rest period for 3 days. After that, the cells, the media was freshened, and then the cells were again grown for another 2 days till day 15. So these day 15 were called as the relapsed CAR-T cells. So further they wanted to check whether the CAR expression. So they found out the CAR expression in both kind of cells. So here we can see in both that 41BB co-stimulatory domain CAR-T cells and CD28 CAR-T cells, the CAR expression was the same. So there was no difference in the gene expression, like CD19 and other gene expression was the same. So this is how they prepared the model. So they named it CD28 D8 cells and 41BB D8 cells. These are the baseline cells, and for activated cells, they have named it D15 cells. So after that, they have checked the whether because there is overlapping in senescence as well as in the exhaustion. So they have checked the markers of exhaustion in these cells. So what they have found out that in there is increase in expression of exhaustion markers like LAG3, CTLA4, PD1, and but after some time, after 6 days, when the bead were removed, the cells were in resting period, the exhaustion marker came to the normal baseline. So that means there was no exhaustion in these cells. But interestingly, we can see there was the persistent expression of TIM3. So it has been found that TIM3 is a marker of senescence. So that means that after this day, the senescence has started in these cells. Then they check the marker of exhaustion in these different CAR-T cells with different co-stimulatory domains, and they have found out that this exhaustion marker was almost similar. So there was no correlation with exhaustion and senescence in these cells. Further, they've
Dr.
studied.
Prerna, sorry
Yes.
to interrupt you. Dr. Shilpa here. Can you just
Yeah.
explain what is the difference between exhaustion and senescence once?
So see, they are almost similar kind of that phenotype is similar, but their pathways are very much different. So in senescence, those cells are metabolically active, but in exhaustion, these cells are not metabolically active. They go in permanent cell cycle arrest.
In exhaustion or senescence?
Senescence. In senescence. In senescence, those cells are metabolically active, but in exhaustion, they are not metabolically active.
Okay, and metabolically active means release of the cytokines.
Release of the cytokines also.
And what else?
Release of the cytokines, and then cell proliferation also. And then there are further markers also available for that. Their activity is very much different, I mean, in senescence.
So,
Cells are active, but they are functionally arrest.
so what we want is when we are inducing the pain, when we are infusing CAR T cells, we want CAR T cell proliferation, and we want to see the we would avoid the senescence. So this what we are
Yes, yes,
discussing is what are the reasons for senescence,
yes, exactly. And
and that is at
another important reason is that is why these days people are focusing on making CAR-T cells in shorter period of time. The longer period of time they are into activation, they lose their activities.
Okay. So when we check for CAR-T persistence, then that time are we able to check what we are checking is only through the surface marker testing, right? Isn't Please correct me. I am asking
Mhm,
very basic questions.
yeah. So sequencing we do by qPCR basically.
Okay, so in that, are we able to assess whether these are senescent CAR-Ts or they are just regular? I mean, they are not. They are functionally active or not? Can we
Yes,
judge?
yes, of course. Yes, of
So
course.
how do we judge the functional activity of CAR T cells?
In further slides, I will show with the markers of proliferation. When we check by qPCR, by sequencing, so there are telomerase length. So there are separate markers for senescence and there are separate markers for exhaustion.
Okay, thanks a lot. Thank you.
Yeah. So in that, though in this, so as I initially also I told that both the domain core stimulatory domains both have different functionalities. So that is why in third generation CAR-T they have used both, because the function of 41BB is different, and when we use separate, so if I say CD28, so it increase the proliferation. Even in this study also they have found out that though the senescence in that 41BB is higher, but the proliferation was good in these cells rather than the CD28 cells. So some functions were good in 41BB cells, some functions were good in CD28 cells, but the end of the result they have found out that these cells which have co-stimulatory domain of 41BB, they go into senescence more faster, and then their rate of proliferation also though increased, but their activity decreased. So in this here they have checked that effector to that T cells ratios. With the decrease in effector to T cells ratios, different definitely the killing ratio is decreased, killing efficiency is decreased. But killing efficiency we can see in comparison to the normal cells, which we call the baseline cell. In comparison to the baseline cell, when these cells are relapsed or reactivated, the killing efficacy was less in those cells, in both the cases, both in 41BB cells and CD28 cells. So that means after this relapse, because they have developed the relapse model, so a cycle of activation and reactivation, activation, it reduced the activities of these cells. So here we can see the killing efficiency is reduced in both kind of model. Proliferation also they have checked by EDUSA. The proliferation is also reduced, which was less in CD28 domain cells, and the proliferation also was less in CD28 cells. So then further, sorry. They produce the in vivo model where they transfer 10 to the power 6 these cells, they transfer prepare the xenograft model. In that they have found out that even the tumor burden also, here we can see these are untransduced cells, these are baseline cells, and these are the day 15 cells. So in these reactivated cells we can see the tumor burden was higher, very much higher in comparison to the normal cells. And then they found out the survival also, the survival rate was also less in case of these activated cells, because they entered into the senescence. That senescence was higher in case of 41BB cells. Then again they were activity also they have checked there. Here we can see that tumor necrosis factor, this release, it was this is again a marker of senescence. So here we can see in day 15 cells, and in case of 41BB cells, that the tumor necrosis alpha was higher in these cases in comparison to the normal baseline cells.
Dr. Prerna, may I interrupt?
Yes, yes, sir. Yes,
Yeah,
sir.
yeah, yeah. So, just for only one request, we are
Yes, sir.
having so many multi-dimensional relation. Okay, one with
Yes,
respect to marker, one with respect to proliferation, expressions, So just wondering, as a layperson, is there any very small booklet to understand the what one-to-one dimension, how one dimension is linked with another dimension, how one is influencing, or could you help us to get some guidelines so it could be easy for
yes,
understanding, at least for me. I'm not
yes, yes.
clinician, so it will be
Yes,
yeah.
of course, sir, because together they have, together multiple things they have checked together. So for me also initially it was confusing that which for what they are comparing for what. So like in this first, they are like here they are comparing the baseline cells like normal CAR-T cell with the cells which goes into senescence, which they have named D15 cells. So these are the normal relapse model kind of. They have artificial relapse model they have developed, which they are checking with the normal CAR-T cells with these senescent cells. So in
Okay,
that,
okay.
yes, so in that these UTDs untransformed cells, which is normal T cells without any CAR-T cells. D3, D8 cells are normal baseline cells, and D15 are the senescent CAR-T cells. So they are comparing these models which are having co-stimulatory domain of 41BB and the co-stimulatory domain of CD28. So they are checking which co-stimulatory domain is better and which is going more fastly into senescence. So in that they are trying to say that these 41BB cells, the CAR-T cells which has 41BB co-stimulatory domain, they go into senescence much faster than the other type of CAR-T cells. So here they are comparing the cells with different parameters. I hope now you a little bit understand what they are comparing.
Yeah, 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 trying.
Yes, yes, so many multiples. Actually, they have, just a moment. Just 1 second, that battery is going. I need to put a charger. Just a moment, please. So after this, they have checked different markers for senescence. In all these, now they checking the cells, CAR-T cells, which are having 41BB co-stimulatory domain, and the cells which are having CD28 domain, the cells which are activated D8 baseline cells with the activated cells. So here P21 they have checked, KLRG1 they have checked, and then these three are the very common senescence markers. So they have found out that this senescence marker, we can see the percentage in 41BB cells is very much higher than the CD28 cells. All three kind of markers were upregulated in 41BB co-stimulatory domain CAR-T cells. Then further they check that together also they have checked two markers together, three markers together, four markers together, these senescence markers all together. They have still found out that the senescence was much higher in case of 41BB co-stimulatory domain CAR-T cells. Later they check the DNA damage also, because DNA damage is also another signal for the senescence. So here also the in DNA, so the DNA damage also we can say, so different genes of DNA damage like H2, AX, ATM and CHK2, they have checked the expression by Western blot. So here they have found out that in case of cells which has 41BB domain, we can see the expression is higher in comparison to the CD28 cells. So that mean again they have concluded that these cells goes into senescence. These cells means 41BB cells goes into senescence faster than the CD8, and the senescence is more prominent in these CAR-T cells. They check telomerase length also. We also know that telomerase length is a marker of senescence. It's shortened. So here we can see in case of that 41BB cells, that telomerase length is much shorter than the CAR-T cells which are having CD28 co-stimulatory domain. Then this is the second model. This was the first model where they've induced the senescence by activation and reactivation. Then they produced the second model, where in second model what they have done, they overexpressed MYC gene. The MYC is a proto-oncogene which is overexpressed in multiple cancer type, and it leads to senescence also when overexpressed. So in this second model, what they have done, they transduced the CAR-T cells with MYC gene. So the MYC gene is overexpressed in these CAR-T cells, and they found out that if MYC is overexpressed, still the senescence marker they have checked, the senescence marker was higher in CAR-T cells which has BB1 4BB1 co-stimulatory domain. So by this model also they have concluded that these CAR-T cells which has 41BB domain, they go into senescence faster, their proliferation rate is low, their cytotoxicity activity is low, and they are not as good as CD28 cells. So in this MYC signaling pathway also, so we know they have shown that MYC is activated in both kind of model. In 41BB, MYC it become dysregulated, leading to senescence. In other type of CAR-T cells, it shows effector function without triggering the senescence. Then they produce the third model where they induce the senescence by irradiating the cells. In this model also the results were similar, that we can see that the senescence markers were upregulated more in 41BB co-stimulatory domain cells in comparison to the CD28 cells. So the proliferation was also higher in case of CD28 cells, the cytotoxicity was higher, and here we can see that T count was absolute. The circulatory T cells were higher in case of CD28 cells. So the main takeaway of this article is that co-stimulatory domain highly impact the function and the fate of CAR-T cells. The co-stimulatory domain both work in a very different way. These domains influence the senescence, persistence, and efficacy of CAR-T cells. Senescence is more prominent in 41BB co-stimulatory domain CAR-T cells. And these CAR-T cells, they have shown that the senescence marker, expression of senescence marker was higher in case of cells which is 41BB co-stimulatory domain. And then, this I wanted to show you later. These CD28 cells, they register senescence better, their proliferative efficacy was better, their effector function was better, and the cytotoxicity was also better than the other kind of CAR-T cells. So in clinical correlation also they have shown that patient sample, they've shown that the cell card to the patient whom they given 41BB CAR-T cells, their senescence signature were linked to poor clinical outcomes, whereas in case of CD28 cells, there were better outcomes were seen in those patients. So this data strongly indicate that the cells which has 41BB co-stimulatory CAR cells, they have impaired functions, they have less efficacy, and then clinical outcome is poor in those kind of CAR cells. Thank you.